If we look at the stages of mitosis now, we have 4 different stages, starting with prophase, then metaphase, then anaphase, then telophase. So the abbreviation that's always known for prophase, metaphase, anaphase, telophase is, we can say, PMAT, to remember the order of them. Now, if we look at each individual stage, starting with prophase. Prophase is divided into early prophase and late prophase. So in early prophase, we see our nucleus, and in our nucleus, we start to see our chromosomes starting to coil up. The nucleolus is still there. And on the side, we have 2 centrosomes. So now a centrosome is a pair of centrioles each. So each centrosome is 1 pair of centrioles that's been replicated. Okay, in late prophase, what we start to see is the nuclear envelope starts to disappear. The nucleolus starts to disappear. And then the centrosomes have started to move to each end of the cell, and each end of the cell we call the pole. So at the top you have the pole, and at the bottom we have the pole, just like the North and South Pole. So they move to opposite poles, and what we start to see is the chromosomes start to coil up and look like chromosomes. Remember, we have homologous pairs, so that's why I've got 2 that are the same and 2 that are the same. Okay, so that is then late prophase, where you start to see all the chromosomes and the nucleus and everything disappears. When the cell starts moving to metaphase, what happens is all of those chromosomes line up in one single layer across our equator. Okay, so if each side is the pole, so remember that's the pole, this is now the equator in the middle, just like the Earth. So what happens is the centrosomes are at each end now, and they make what we call spindle fibers. And those spindle fibers are made from microtubules, and each spindle fiber goes through each chromosome from one pole to the other, so that those spindle fibers are holding each chromosome in the middle from top end and bottom end. Okay, so we recognize metaphase by all those chromosomes lining up in the middle, so you end up with this dark line that you can see at the equator. So once they're all lined up and the spindles go through each of them, then the cell goes through anaphase. So in anaphase, you now have your centrosomes at each end with the spindle fibers coming out. But what happens is those spindle fibers each contract back towards each end of their centrosome. And when they contract, they take each chromatid from each side with them. So it always forms this V shape because it gets pulled by its centromere in the middle, and that one, and that one. So each one gets pulled away from its pair. That's, remember, genetically identical because each chromatid is the same, and it pulls to opposite poles. So at anaphase, what we see is 2 separate lines moving towards each side. And then at telophase, what happens is the whole cell starts to cleave inwards, like we say, in the middle, and that is called cytokinesis. So where the cytoplasm starts to pinch to start to make two separate cells, we have the nucleus starts to reform. And inside each nucleus is one single chromatid from each chromosome. So we start with the same number of chromosomes as we end with in each cell. They're just now single chromatids as opposed to there where they were whole chromosomes. And each end has its own centromere, and we start to reform the nucleus. And inside there is a division of organelles as well. So we have all the same organelles as before, the ER, the lysosomes, all those things in there and in there. We just now have two separate cells at the very end. Okay, so those are our stages of mitosis. And I just want to show you specifically here where the centromere is. That is because in the chromosome as a whole, we've got our one side and our other side. And remember, I told you this part in the middle is a centromere. But inside that centromere, on the one side over there and on the other side over there, we have what's called a kinetochore. So a kinetochore is a protein that binds to the DNA on each side, and it allows the spindle fiber to actually attach to it so that it goes through. So when it pulls, it's getting pulled by this protein over there that's moving away to opposite sides. So that's how it makes it easier to pull the whole thing apart, because we have those little proteins called kinetochores. Okay,